Hi students, today we will be looking at nutrition. So basically we divided up into 4 different segments. We will have carbohydrates, proteins, fats, and lastly the component of water and minerals itself. Okay, so let's get started. We'll look at carbohydrates first. So carbohydrates, they have got main elements will be carbon, hydrogen, and oxygen. In fact, when we look through the other 3 different nutritions, we have got your proteins and your fats. They will also consist of carbon, hydrogen, and oxygen, which are the main components or the main elements that make up majority of our nutrition. Okay, generalized formula is this. Okay, CnH2nON. So when we look at glucose, the most common type of carbohydrate, C6H12O6, we can see that it fits into this generalized formula. Okay, so carbohydrates, they are the main source of energy that is needed by the human body. So the sources over here, I mean the functions of this, so main source of energy here. It is also used as a supporting structure, for example, in cell walls of plants where they exist as the cellulose itself. So cellulose is also a main type of carbohydrate, a complex carbohydrate, or otherwise known as a polysaccharide. So excess of these carbohydrates can be converted into amino acids and fats. Some may be used to form nucleic acids, which is part of the DNA component. So which is why carbohydrates, they can be the starting point to manufacture other substances. In plants, carbohydrates are also used to produce nectar. So nectar attracts insects to come to the plant itself, to the flower rather, and it helps in the process of plant reproduction. Now, 2 ways to test for carbohydrates. One is for starch itself, the other one is for reducing sugars. So to test for starch, you'll be using things of your iodine solution. So a few drops of the iodine solution onto starch, like for example that is contained in your bread or potato chips or potatoes, it will change from brown to a blue-black color, which is a positive test for starch. Okay, now reducing sugars is used for your disaccharides and your monosaccharides. So all your disaccharides and monosaccharides except sucrose will test positive. Okay, so except sucrose, it will test positive for reducing sugars here. So over here, you have got your blue color solution, okay, which is the Benedict solution mixed with copper 2 sulfate, will test positive for reducing sugar and produce a red precipitate or brick red precipitate. Now, for this test, you can't really see exactly how much is present, but you can roughly tell how much of the sugars are being present, and which is why it is called the semi-quantitative test. So all these different colors in large quantities, a brick red solution is, or brick red precipitate is being produced. Now for IP students, you will be doing the test for sucrose, which is a non-reducing sugar. So we can't use the Benedict's test. So in this case, you will be using hydrochloric acid, hydrogen carbonate, and finally the Benedict solution here. Okay, so over here, you hydrolyze the sucrose into the main 2 different types of monosaccharides, which is your glucose and fructose. So glucose and fructose, they both come together to form your sucrose over here. Once it is hydrolyzed into these 2, then it can be carried out to use for Benedict solution. So how do you hydrolyze your sucrose? You boil it with hydrochloric acid for a few minutes, and then this acidity has to be neutralized by adding a small amount of hydrogen carbonate. So hydrochloric acid is acidic, of course, and hydrogen carbonate is slightly alkaline. So this will neutralize each other, and you know that the reaction has been completed because when there is hydrochloric acid present, it will react with the hydrogen carbonate to produce carbon dioxide gas. But once the reaction is completed, meaning to say that there is no more hydrochloric acid, therefore there is no more carbon dioxide present, which means effervescence stops. Okay, so by then you will be able to hydrolyze your sucrose into glucose and fructose, and you can test for reducing sugars using your Benedict solution. Okay, so you can tell that you have got different classes of your carbohydrates here. So the main few classes of your carbohydrates can be classified into 3 main groups. The most complicated form is your polysaccharides, which is why it's poly. Now, polysaccharides can be broken down into disaccharides, and the simplest units, di means 2 of it, So it can be broken down into simpler forms, which are your monosaccharides, which are just single. So monosaccharides, we have got three different kinds: glucose, fructose, and galactose. Okay, now glucose is the one that humans use in the easiest form by the brain, by the cells itself. Now any of these two can come together to form my disaccharide through a combination or reaction called the condensation reaction. So when I've got two small monosaccharides coming together, it will undergo the loss of a water molecule, and this loss of the water molecule is known as the condensation reaction. Okay, so over here I have got two glucose coming together. This bracketed form, the water is being lost, and now I have got a maltose, which is a disaccharide. So two monosaccharides coming together to give me a disaccharide, and is joined by what we call the glycosidic bond. Okay, now glycosidic bond is more for IP students, so those doing pure or combined don't have to worry too much about this. Now, if let's say I have got a maltose and I want to break it down into glucose, okay, to be digested, this can also be done. So it's a reversible reaction with a suitable enzyme, but the opposite reaction, you have to add back a water molecule. Okay, going from left to right, I have got the loss of a water molecule. So it only makes sense that if I go from the right to the left, I need to add back a water molecule. And this addition of a water molecule is called a hydrolysis reaction or hydrolytic reaction compared to a condensation reaction. Because hydro means water, so addition of water in the reverse reaction will be hydrolysis. Okay, so types of disaccharides, I have got three different kinds. You can see that it comprises of any two of the monosaccharides here. So we have taken a look at glucose and glucose, which will give me maltose. Okay, now glucose and fructose give you sucrose. In plants, when you have got photosynthesis, often glucose is being produced. Okay, now glucose will then combine with fructose, and the glucose in plants is converted to sucrose, which is transported around the plants, which is why sucrose is the common form for transportation, and you can find that in sugarcane stems or in sweet fruits. Okay, sucrose is for plants, we don't have it in mammals. Okay, now lactose is glucose and galactose. Okay, so that will get you lactose, and of course, because it is a condensation reaction, you have got water that is being produced for all of this. Okay, now this is just for illustration. So monosaccharides combine to get you your disaccharides. Okay, now disaccharides can then be combined to give you your polysaccharides. Okay, the main one that we'll be looking at is for glucose itself. Now in humans, excess glucose can be stored as glycogen here. Okay, so in animals, in humans, glycogen is being stored in livers and muscles. Okay, in plants, we have got cellulose and starch. Starch is often found in your plant areas, things like the potatoes or the roots itself. Cellulose is often made up as part of the cell wall for the plants, and when we eat it, it becomes a source of dietary fiber for humans. Okay, which is why it's best to eat apples unpeeled rather than by peeling it. Now, the next part we will be looking at is fats. Okay, so we'll come back in a while.